Hello, welcome to my presentation. I'm Priyal D'Souza. I'm going to talk about clinical genomics. So the subtitle of my talk is the basics and an up-to-date technological perspective of clinical genomics. So what is clinical genomics? Genomics is a bottom-up approach that uses DNA sequences to identify genetic changes and which result in molecular mechanisms likely to cause disease. It is one of the most common ways now. Has been gaining momentum, and it's very, very fast. So it's going to be the future. There are several types of genomic DNA-based genomic analysis, and the one I'm going to be talking about is really the last one here on the list, which is the targeted NGS panels, which are used in clinical diagnostics. Historically, the first genomic tests were based on single gene amplification, sequencing, and mutation. And they have been around since the late 1980s. This was followed by microarray DNA hybridization panels, which were used from the beginning of this century. And more recently, in the last decade, these four techniques rather, have been gaining momentum and used in research and development labs for diagnostics. These are namely the whole genome, the whole exome, and ChIP-seq and methylome, which are, as I said, mainly in the R&D arena. However, the targeted enriched panels, which I'm going to talk about, is becoming more commonplace in clinical diagnostics. It can still be used in an R&D setting, but the main use is for clinical diagnostics. And that's been possible since about 2013. So targeted enriched NGS panels they're an off-the-shelf end-to-end diagnostic solution. They can be they're normally disease-specific, and they have validated assays which meet regulatory accreditation. So what is an NGS panel? Well, basically, it's a series of regions in your genome which can be amplified simultaneously in one tube or at most two tubes. So these regions are basically previously identified predetermined regions which contained mutational hotspots which are likely to cause disease. And in a panel, there are thousands and thousands of these sequences spread right through the genome. Here they're showing 4 different regions, basically pretty close to each other. That does not need to be the case. They can be scattered all over the chromosome, or it could be or spread out through all different chromosomes. So an example of some NGS panels are ones used for bladder cancer, for diseases like renal cell carcinoma, breast cancer or BRCA, and also hepatocellular carcinoma or liver cancer, if you like. And these panels normally consist of somewhere between 15 to 50 genes, which are likely to contain mutations that might be giving rise to a disease. So what's actually involved in getting results for diagnostics? Well, you start off by making a DNA preparation from either a blood or a tumor sample, and then you follow this workflow. A scientist would normally do this. A clinician doesn't do this unless they're trained to do it, of course. And they would normally, the scientist will start by constructing a library of DNA molecules and preparing the template which goes on the sequencer. And the sequencer will once it's loaded, it will process the sequences and generate lots of electronic sequences which get uploaded to a large computer on a server, and it starts analyzing the data and finally results in generating a final report for the pathologist. So what's involved in this process? Basically, the clinician gets this report, and normally what happens is the clinician asks the pathologist to review the automated report and generate a diagnosis according to the mutation data produced. That's it. Thank you.